Dr. Gail Volk, a plant physiologist with USDA ARS at the National Laboratory for Genetic Resources Preservation in Fort Collins, Colorado, USA. I, along with Dr. Robert Kruger, also with USDA, and Dr. Fred Gmitter with the University of Florida, led the effort to develop a global strategy for the conservation of citrus genetic resources. It was spearheaded by the Global Crop Diversity Trust. The goal of the global strategy for the conservation of citrus genetic resources is to provide a framework for the efficient and effective global conservation of citrus crops. Citrus is an incredibly important crop, whereby oranges, mandarins, lemons, limes, grapefruits, and pomelos are produced in tropical and subtropical regions around the world. Citrus faces serious threats due to multiple diseases, especially citrus greening, which is also known as Huanglongbing, or HLB. Ensuring that breeders have access to novel citrus genetic resources that have resistance to citrus greening and diseases caused by other pathogens is critical for the development of new cultivars and rootstocks. Prior to the global strategy, there weren't any comprehensive reports summarizing the availability of citrus genetic resources in gene banks around the world. The strategy was developed through a process that identified and then engaged with citrus gene bank collection managers from around the world. First, lists were compiled with contact information for citrus gene bank collections. Then a survey was developed and then released to gene bank managers and curators. The survey requested information about citrus collections relating to their ownership, composition, availability, security, cleanliness, and challenges for their maintenance. Citrus is an Annex I crop for the International Treaty on Plant Genetic Resources for Food and Agriculture, so it was particularly relevant to learn about possible availability of citrus plant genetic resources from countries that have ratified the plant treaty. Data were compiled and then summarized. Based on the information from the survey, a global system was described that proposed how citrus conservation could become more coordinated, systematic, and unified. A draft strategy was reviewed by the survey participants. The strategy was then reviewed by the Crop Trust and finalized and made publicly available. Ideally, the diversity of citrus and its wild relatives will be conserved and available in a disease and pathogen-free state in perpetuity in a secure, distributed network of gene bank collections that provide collection data, such as passport, phenotypes, and genotypes, in a standardized common information system. Large collections, such as the ones in Brazil, China, the US, Japan, France, and Australia, possess a wide range of cultivars, land races, and wild species. Other collections may specialize in local cultivars that are not available anywhere else. One key concern is that most collections are not secured in a secondary location. Aside from cryopreservation efforts at the USDA National Plant Germplasm System, where citrus cultivars are stored as shoot tips in liquid nitrogen. Citrus is used for breeding, resale, research, as well as molecular characterization efforts. Some collections are generally available within country, while others are available at an international scale. If funding was available, the following priority actions were identified in the global strategy. 1. Increase citrus gene bank community cooperation by establishing an international working group and developing a citrus community information system. 2. Support data collection and documentation efforts for citrus collections. 3. Identify taxonomic gaps, both within cultivars and related genera, in citrus collections and fill those gaps through collections and exchange. 4. Increase citrus collection health and security, particularly for collections that have vulnerable, unique plant genetic resources. 5. Provide training opportunities for the citrus gene banking community on a wide range of topics through a combination of affordable in-person and online options. And 6. Develop, maintain, and distribute materials from clean, secure, international citrus collections at one or more locations that capture taxonomic and the genetic diversity of citrus. All of these activities are directly related to plant treaty efforts to increase the quality of plant genetic resources collections and to make them more available. In particular, many countries with citrus collections have ratified the plant treaty, thus making their citrus collections available to international research and breeding programs after acceptance of the Standard Material Transfer Agreement, or SMTA. If additional resources were available, then citrus collections could be genotyped and phenotyped, thus allowing for collection comparisons and improved identification. Furthermore, it would increase the value and utility of the collections. The development of new disease-resistant citrus is dependent upon access to quality citrus collections in GeneBix.